Hi, everybody, and welcome back to our channel, or welcome. Today, we're going to be learning the cell biology basics, and I'm your presenter, Kim. So, what is cell biology? What are cells in the first place? So, all living beings are made out of cells. They are the most fundamental units of life, and to every cell, there are four features: a cell membrane, which you can see here as this yellowish orange. The next feature is cytoplasm. Cytoplasm is like this watery substance that fills cell membranes and they create the needed internal environment of a cell. So genetic material, meaning DNA or RNA, exists in all living beings. And for animal cells and plant cells, the main genetic material is DNA, but RNA also exists. In bacteria cell, it could be RNA, DNA, or both. And then we have ribosomes. So ribosomes are like these little dots scattered across the cytoplasm. There are many of them because they're very important. They're the site of protein synthesis. Protein synthesis is very important because being able to make proteins, which serve a lot of functions to the cell, is critical for the cell survival. We'll go into depth about protein synthesis and the function of proteins in another video. So cell biology examines the structure and functions of cells. And in this series, we're going to focus on internal cell structure and processes. So here's the mind map explaining the topics we're going to cover. First, in this series, we're going to cover biological molecules, enzymes, transport in cells and cell membrane. But first, let's start with biological molecules. So there are four groups of biological molecules: proteins, carbohydrates, nucleic acids, and lipids. Note that these are polymers, meaning they're made of smaller units called monomers. So proteins are made of amino acids. Carbohydrates are made of monosaccharides or simple sugars. Nucleic acids are made up of a unit made up of nitrogen base, pencil sugar, and phosphate group, or are commonly known as a nucleotide. The lipids are made out of glycerol and fatty acids, which sort of count as one unit. So some examples of proteins, we have enzymes, we have receptor proteins and antibodies. So antibodies you may have heard of. They are in vaccines. They are what helps us fight infections and bacteria and virus. And receptor proteins, they're sort of like antennas on cell membranes, and they're involved in cell signaling, which we'll cover in another video as well. For carbohydrates, our main interest is glucose because glucose is the primary substrate for respiration, meaning it is the primary form by which our body sort of gains / extracts energy. So for nucleic acids, we have DNA, RNA, and like mRNA, RNA, tRNA. I know it sounds Greek, it sounds very weird, but notice how it's all blue. Everything in blue means it's important, and therefore will be covered in some point of the series. And this entire cluster is blue. So we'll have a separate video on DNA, RNA, cell division, all that in our next video. So look forward to that. And then we have lipids, or commonly known as fats. So lipids are very important to us. They may have like a bad reputation in the diet culture and everything, but they make up very important double layer components in our cells even, such as cholesterol and phospholipids. Steroids are also lipids. So steroids are hormones, but they're very important hormones such as estrogen and testosterone. So first we're going to be looking at enzymes. So enzymes, some things to know before we formally start talking about enzymes. So enzymes are proteins. Proteins are made of amino acids. There are four levels to a protein: primary, secondary, tertiary, and quaternary. We usually say this is the primary structure, this is the secondary structure, the tertiary structure, and the quaternary structure. So in the primary structure, we have amino acids, which are these little colored circles joined together by a peptide bond, and they form this really long chain, and that is, we can just call that a protein. For the secondary structure, this chain of amino acids, they sort of arrange themselves into either a helical shape or a sort of like a folded shape. We call this a alpha helix and beta beta sheet. So these two structures are held together by hydrogen bonds. In the tertiary structure, we have multiple alpha helixes in yellow, multiple beta beta sheets in blue, and they're also again folded in to sort of create this three-dimensional shape. They're held together by four bonds: ionic bonds, disulfide bonds, hydrophobic interactions, hydrogen bonds. Let's pause here for a minute. Doesn't matter the bonds, doesn't matter what bonds. Why are the bonds even there in the first place? Very good questions. First of all, bonds matter, and the type of bonds that form depend on the amino acid sequence, like the sequence of amino acids in the primary structure. So instead of having this amino acid, I swap it with this amino acid, and then the primary structure, the sequence is different. And because of the change in the primary structure, there will be a change in the tertiary structure because the bonds come from the unique interaction between the amino acids and its neighbor. And if that arrangement is changed, the bonds at tertiary level will change. And that is very important to an enzyme. We'll see why in a bit. So at quaternary level, we just have multiple amino acid chains. We have multiple polypeptides. In the tertiary structure, it's still one long polypeptide. In the quaternary structure, it's multiple polypeptides. So, what are enzymes? So they're biological catalysts that speed up reactions. And all enzymes are proteins, but as I've seen in the previous slide, not all proteins are enzymes. So enzymes function due to their specificity. And their specificity comes from their three-dimensional shape. So the tertiary bonds we talked about earlier, so they form this 3D space filling molecule. So, and like the gray area here is called the active site. It's where the substrate sort of comes in, like how a car comes into a garage. It's like place for the substrate to sort of dock. Enzymes and substrates, you can think of it as a lock and a key. Only the correct lock, only the correct key would be effective on the lock. So the shape of the lock, the key, they must not change. Like if you have the same lock and then you have the same key, but then you go out and like it gets smashed or something, and like on a car or something, that key will no longer fit into the lock, correct? Then it's no longer useful. That in the same way, when the bonds here, the tertiary bonds change, enzymes, their shape changes. Therefore, the reaction it usually catalyzes, it can no longer do that job. So when these bonds change, which can be either of like really high temperature, really low, or high pH, or the amino acid sequence gets reordered and it becomes an entire new sequence, then these bonds will not be the same. Therefore, this active site cannot form. Therefore, the enzyme cannot function. It cannot do its usual job. When the shape of the active site changes and the enzyme no longer functions, we say the enzyme has denatured. But first, let's look at how enzymes function under normal optimal conditions. So this is an enzyme. This gray area is the active site. Boom, a substrate has locked into this active site. You can see it's a perfect fit. And you can see this pink little streak here. That is the bond that this enzyme is going to be catalyzing. It's going to be breaking that one. And then boom, the products are formed. They're like two parts of the same thing. And now that the products are formed, where do they go? Like the reaction was catalyzed, the enzyme was involved in the first place because we wanted these products, right? But where do they go? How do they even go where they need to go? And here comes in transport in cell membranes. So there are four main transport processes: diffusion, osmosis, active transport, and facilitated diffusion. So diffusion is probably the one you're most familiar with. Whenever you put in like a drop of food coloring into water, you can see it just spreads. Even if you don't mix, you don't do anything to it, given enough time, the entire thing changes into an even shade of red. It's not even a gradient. At some point, it's a gradient in terms of color, but after some time, it becomes an even shade of red. We can say the food coloring or the substance has moved down its concentration gradient. So osmosis is just diffusion, but exclusively for water. And instead of saying concentration gradient, we say water potential gradient. It moves from a region of high water potential to a region of low water potential. So active transport is the reverse of diffusion. Diffusion is something that happens naturally, as how a ball sort of falls down a hill, given that there's no friction. Active transport is a ball going up the hill, going against gravity. But in molecular terms, we say the substance goes against its concentration from low to high concentration using energy, because a ball wouldn't be able to go up a hill unless you give it like rockets or something. And in cells, that energy is provided via ATP. Facilitated diffusion is when a substance moves down its concentration gradient via a channel protein or carrier protein. So facilitated diffusion is something that is sort of limited to cell membranes because channel proteins and carrier proteins are embedded proteins in a cell membrane, and I'll show you what they do and what they mean in just a second. So here is what I was talking about earlier, a ball falling down, substance moving down its concentration gradient, that's diffusion. Next we have osmosis. This is a water molecule, and yeah, it just goes down its water potential gradient. Next we have active transport. So the substrate is originally at a region of low concentration, and then we give it energy through ATP, and then like a rocket, it sort of pushes the ball up. It's like a car engine, it just gives the energy needed to get you up a hill. So facilitated diffusion. So here's the interesting part. Okay, so first you saw how the molecule it fell, but then it stopped because there is this layer here, this border here. So that is the cell membrane or some form of membrane. It doesn't have to be the cell surface membrane. It just be a membrane, and it blocks the substance from entering. So the substance has a channel protein that is specific to the shape of this substance and allows it to pass through, just like that. So this is a channel protein because it's embedded, doesn't move, and it like lets it pass through. If this is a carrier protein, think of it as like this little tunnel ending here. But this substance get into the tunnel, but then the tunnel itself moves. So again, it's like a car. You get in, the car drives, you get out, you're where you want to be. So facilitated diffusion does not require energy because the substance is simply moving down its concentration gradient. So let's talk about cell membranes. So first, let's know two things. So lipids, which I was talking about earlier. So this is the glycerol unit and the three fatty acid chains it is attached to. So lipids are insoluble in water. So next are phospholipids. So phospholipids are made of one glycerol molecule attached to two fatty acids attached to a phosphate group. The phosphate group gives the phospholipid its unique property of having a hydrophilic head, meaning a head that likes to interact with water, and a hydrophobic tail, which means it does not like to interact or mix with water. So this is what a cell membrane looks like. So we have the phospholipids lined up, and then we have the phospholipids lined up in another layer again, but in an opposite direction. So what this creates, sort of a middle layer. So this, because of these two layers of phospholipids, this will be a bilayer. And because of the middle layer being solely of hydrophobic tails, it makes the cell membrane impermeable to anything that is water soluble, including water. And water has its own channel which it goes through, which is called aquaporin. So let's say on this side we have tissue fluid. On this side we have the cell cytoplasm. So why do we have this arrangement of tails facing inwards, heads facing outwards? Because the heads, they like water, they interact with water to form bonds which stabilize the entire membrane. And the tails, they prevent anything water soluble from entering. So if a substance must diffuse through, it has to go through channel proteins or carrier proteins. And if a substance is not needed, if it's not wanted, the cell will not make those channel or carrier proteins and it will not be let through. So the cell can sort of protect itself and it can regulate what enters and exits itself. And as we can see with the previous example, the substance which must undergo facilitated diffusion has a channel protein, a carrier protein that is specific to it, meaning the two shapes are complementary to each other. Let's say this is a cube box, and then this will have a rectangular tunnel so that only this molecule and only this molecule can pass through. Of course, in real life, the shapes are very complex. They're not even the shapes we see in every day. If you're interested, you can sort of like Google it and just look at it like 3D structure of proteins, enzymes, channel proteins. Yeah, knock yourself out. Okay, so let's turn our attention to receptor proteins. So with the receptor protein, you know for the receptor proteins, you'll notice that there's like this little antenna that's sticking out, and this is the part that the molecule binds to. So receptor proteins are involved in cell signaling. They are the cell surface markers, and only the specific molecule can bind. So let's say this is an insulin receptor. Then only insulin can bind to this. And when that binding happens, it can trigger a cell signaling cascade. So yeah, we've covered all three topics. Yeah, thank you for watching and see you in the next video.